I'd like to welcome everybody to this webinar. I'm going to discuss today our experience with our phase 1 trial of spinal cord transplantation into patients who are suffering from ALS. This trial is called the NeuralStem trial because NeuralStem is the company that actually owns the cells and is supporting this trial. So thank you very much for everybody for participating and for listening. Just to disclose my funding, I do get funding from NeuralStem specifically for running this trial, though I have no investments. I get funding from the NIH, specifically the NINDS and the National Institutes of Aging, the ALS Association, and the Muscular Dystrophy Association. I also have many collaborators. This is not a trial that can be done by myself, and pictured here is Nick Poulos. Nick is the neurosurgeon who really invented the procedure for injecting stem cells into the spinal cord. Nick is the neurosurgeon here and is a close colleague. Also, we have a group of investigators here, nurses and other scientists here at Emory that help run this trial, along with pathologists and molecular geneticists helping us to find the cells in the spinal cord. At Michigan is Eva Feldman, who really started this whole trial off. We call her the mother of the trial, and she started it with NeuralStem several years ago with her idea. So at Rut Cove up at the Beth Israel Hospital, David Senator helps us again with the identification of cells, and again, the folks at NeuralStem who are intimately involved in this project. ALS is a fairly devastating disease, and to those of you who are listening, you probably know something about it. But it can take relatively young, healthy people in the prime of their life, and in fact, cause them to become severely disabled over a relatively short period of time. Here are 3 examples of patients of mine over the last several years who have gone from being relatively independent with a new diagnosis of ALS to in wheelchairs and on ventilators in order to survive this horrible disease. ALS is characterized by weakness and atrophy in the hands, as you can see here. And in this video that is kind of staggering along, you can see the abnormalities of the tongue and the twitching motions that you see in the muscle that we call fasciculations. These are relatively pathognomonic of ALS. And this is what we see in patients who come into the clinic. It's not that we haven't been trying to cure this disease over many years, and many investigators before me and many investigators after me will continue to work trying to find both causes and cures for this disease. And that's one of the reasons we're moving toward a new type of intervention, which is the stem cell trial. But here's a list, a very actually abbreviated list of drugs that have been tried in major clinical trials for ALS, and all the ones in red here are ones that have been tried and failed. The only one that has really survived is this drug called Riluzole. Riluzole was identified and tested back in the early 1990s and was approved by the FDA in 1994. It is still the only FDA-approved drug for ALS. There are some symptomatic treatments, including this one called Nudexta or dextromethorphan-quinidine, which helps with some of the emotional changes that can occur in patients with ALS. And the ones in blue, tamoxifen here and the troponin activator, which is part of the benefit trial, which is going on now, are still being tested but have not yet been proven to be helpful. So we move towards stem cells. Why stem cells? Well, stem cells are magic. Many patients, many investigators, and the world at large think that stem cells will be helpful for many different diseases. At least in the field of neurodegenerative diseases, and specifically ALS, we just don't know the answer to this. And until we figure out whether stem cells can be therapeutic for patients with ALS, we need to do rigorous trials. As many of you out there know, there are, quote, stem cell treatments, close quote, that are being offered usually for cash around the world, but these are not well regulated. There are no data coming out of most of these trials, and it's not clear whether they work or they don't work. So our goal here was to actually design a true clinical trial to see whether we can, number one, do this safely, and number two, whether we can make a difference in the course of this disease. The stem cells that we're using come from a fetus, a single fetus, generated in the early 1990s, and Carl Joe, who's the principal and CEO at Neuralstem, really made his career on developing a way to propagate these cells so they could be used for human treatments. These are called neural stem cells, and neural stem cells mean that they can really only make neurons and the other cells of the nervous system, the glial cells. And so, in reality, as opposed to embryonic stem cells that can make all types of cells, including tumors, these cells we don't think can make tumors and probably can't make cells other than nervous system cells, which is a positive thing in terms of treatment of nervous system diseases. Why do we choose ALS? In fact, Neuralstem could have chosen these neural stem cells to choose to treat many other diseases of the nervous system, but we think that this is a good choice to give a first trial. And it's because these are mature patients who are able to provide true informed consent. That is, there are a fair number of dangers and risks associated with this disease, and so we want to make sure that the patients who are undergoing this treatment understand what they are, they're mature, and they can actually make an informed consent decision. It's an adult onset disease, again, so adults can make these decisions, and it's got a relatively short course. And why is that important? Well, the relatively short course allows us in real time to identify cell survival and the neuropathology in real time. So if you think about it, if you injected these types of cells into young men who have had spinal cord injuries that might live another 30 or 40 years, you might not find the answer to whether your cells have survived or you've caused any neuropathological damage for a very long time. Similarly, a disease like Alzheimer's disease, which can have a course of 10 or 12 years, many of the investigators who start off in that trial might not be around anymore when those patients come to autopsy. So we think ALS is a good choice. There have been many preclinical studies in patients, in animal models of ALS. And this is just one of those studies that is published from the Neuralstem group, and this is in what's called the SOD1 animal model. This specifically is a rat, and the SOD1 model is a rat that carries a human gene mutation in the gene for SOD1, or superoxide dismutase 1, which we know in humans will cause disease. And interestingly enough, animals who carry these human mutations develop a disease that looks like ALS, and so they're used as models of the disease. And so what you can see here in the red, animals injected with the live cells, neural stem cells tend to live longer than the animals injected with the control cells or dead cells. And you can see this here, that the motor neuron number is more in the animals that have those live stem grafts versus the control grafts. And even in the picture here, you can get a sense that these purple motor neurons are more prominent here than they are in the controls. So that's just one of the small pieces of data that supported the idea of moving this toward humans as a therapeutic trial. But also we needed to figure out how to get these cells into patients. And so Nick Poulos, over the last 10 years or so, has been working on this problem in terms of developing therapies to the spinal cord, and developed this apparatus which we use in the operating room now. He used them first and tested them in pigs, and then went to humans to show that we could very accurately and reproducibly inject stem cells into the region of the spinal cord that we thought needed to be there. I'm going to show you here a bit of a movie, and this, again, is a movie of some of the preclinical work with the pigs. And the mini pig is actually a very good model for testing spinal cord therapies in humans, because the pig spinal cord anatomically is very, very similar to the human spinal cord in size and location. And so getting it right in the pig is probably the best way to figure out whether we can get it right in the humans. And what you're going to see here is that Dr. Poulos is opening up the coverings of the spinal cord called the dura, and you can see there the spinal cord underneath, and the pump is being filled with the cells, and then that pump and the needle that is used to inject into the spinal cord will be attached, as you can see here, to the apparatus, and this will allow the surgeon to very accurately place that needle and inject these cells into the spinal cord. This is some of the data. There have been more than 100 pigs, probably closer to 150 pigs now, operated on by Dr. Poulos. And so this was all perfected before we went to humans. And this is just an example of one of those studies. This over here is the Tarlov scar, and that the higher, or 4, means the animal is perfectly fine. As it goes down, the animal has difficulties with things like moving their hind legs or getting up from a seated position. And you can see here, for the first few days after operation, in these 3 different groups of pigs, you can see here that some of the pigs had some difficulty, and that's probably had to do not necessarily with neurological dysfunction, but with the pain associated with the surgery. But very quickly, over the first 1 to 2 weeks, all of these pigs tend to recover, and they do quite well out to the 60 days that they're allowed to survive. We can also see, at least in the pig here, that we can mark the cells that have been injected with a specific marker for human cells. And there are specific markers for human cells that can separate them from pig cells. And you can see here that Dr. Poulos has very accurately injected these human cells into the pig spinal cord, into the region of the ventral horn, VH, which is where we want them to be, which is where the motor neurons live, and those are the cells that we'd like to eventually protect. And so we need to move to humans, and these were some of the concerns we had when we started off. Number one, what's the long-term potential toxicity of these stem cells? Is it possible that they could form tumors, even though we think they don't? And could they spread from the site of delivery? And that might be a good thing or a bad thing. If they spread from the site of delivery and they're good for you, that might be a good thing. But if they spread and they're bad for you, that might be a bad thing. And we can't get these cells out once they've been put in. Number two, there's always a potential for catastrophic outcome. So patients with ALS have horrible disease, and we know that they're getting worse. But we could potentially damage their spinal cord and make them even worse than they are. And so we could somehow get in the way of their quality of life. The majority of patients don't have pain. We could create pain. And the majority of patients don't have problems with control of their bowel, their bladder. And again, tinkering with the spinal cord could create those problems, which we don't want to do. So we had this worry that we could create a catastrophic outcome. But we also had to follow these necessary steps in order to get FDA approval to move forward. And again, characterization of the stem cell product, which was done by the NeuralStem group before going to humans, the toxicology in animals, some of which I've shown you in both the rat and in the pig. There has to be some evidence of efficacy in an animal model, and at least in the rat model we showed that we could make some difference, at least in terms of lifespan of the animal. And then we need to develop a protocol that was focused on safety, and that was what our necessary step was once the FDA said we could do this. And so our goals for this phase 1 trial were safety, safety, safety. The idea was, is this a safe procedure? We do not want to move ahead with stem cell injections into the spinal cord if we actually could potentially cause harm to these patients. And so we were interested in safety of the surgical procedure. Will these patients tolerate this invasive procedure, and could we make them worse? The other interest is that if we could prove safety, that would allow us for a rapid progression to a phase 2 trial, where we could then escalate the dose and potentially start looking for efficacy to see whether this changes the course of disease. So we had 5 treatment groups in that phase 1 trial. This was called a risk escalation trial, meaning that we started with a group that we thought had relatively small risk and moved to a group that had relatively high risk. The low risk group we thought were patients who could not walk. And so since we were injecting into the lower spinal cord that affects and controls the movement of the legs, if we started out with patients who couldn't walk, at least we couldn't make them non-ambulatory, unable to walk with any damage that we caused to the spinal cord. And we first started with unilateral injections into the spinal cord down there, and then after those 3 patients, we went to bilateral injections. After that, we moved to patients who could walk, and we did unilateral injections into the lumbar cord, and then group C was bilateral lumbar injections. Again, moving up with each group of patients with either the intensity of the treatment, which is either 5 injections or 10 injections, or the relative protection of the spinal cord in patients who did not have as much disability as the patients before them. So as we move forward, these patients had less and less disability. And the group D, actually, we started out going up into the neck. And again, we're concerned that injection into the neck could cause much more problems than down into the lower spinal cord, although this is our ultimate target, because the motor neurons in the neck or the cervical spinal cord are the ones that control breathing. And our ultimate goal here is to maintain breathing in patients so they stay alive. And eventually we went to patients who had both injections into the lumbar spinal cord and the cervical spinal cord in 2 surgeries. This is the way we target the spinal cord. We take MRI scans, as you can see here, looking specifically at the region of the spinal cord. These can be measured, the region of the spinal cord, and then specific injection needles can be designed specifically for these patients to go to the depth that's necessary to try to hit the lumbar or the cervical motor neuron pool, which sits down here, and we go in through here, called the dorsal root entry zone, through the spinal cord to here. And here's a picture of what these needles might look like. And as we move forward, we use something called an injection pump, and this injection pump controls the rate of injection. Again, we don't want to create too much pressure in the spinal cord, and so we want to have a very slow rate, and we only inject 10 microliters, just a few drops of these neural stem cells, and that is done over about a 2-minute period. Here again is another movie. This again looks very much like the pig movie, but it is not. This is one of our injections into a human patient with ALS. And what you can see here as the movie moves along, which it looks like it's not going to do, I apologize for that, but what we're seeing here is the human spinal cord, and the human spinal cord is being opened. This is the surrounding connective tissue called the arachnoid, and you can see the blood vessels on the surface, and eventually this needle here will be placed into the spinal cord itself, and the injection will take place. So here are the outcomes of our phase 1 trial. We had 15 patients and 18 injections, and that is because 3 of our patients had 2 surgeries. They had first lumbar surgery and then cervical surgery. We can see here the age at surgery was very varied, with a range of 35 to 66, and the range in terms of the duration of disease was from 1.5 to a few patients with very chronic disease out to 12.5 years. And this has all been published. The first 12 patients at least were published in the journal Stem Cells here in 2012. And we've had 6 deaths thus far in the surgery. All of these deaths were due to the progression of ALS. None of them, as far as we're concerned, were due to either the injection or the surgical procedure itself, or the spinal cord stem cells that were injected in. And you can see here there are some patients who lived a very long time, 30 months after surgery, and the one woman who was injected died 7 months after surgery. These are measures of progression of disease, and again, this is a safety trial, and so what we were interested in was whether the course of disease would change, and so we followed these patients prior to surgery and then after surgery to see whether the rate of change would change following surgery. And what you can see here in some of these patients, patients 7 through 9, patients 10 through 12, and patients 13 through 15, and what you can see here is that the slope of decline is about the same preoperatively. The line is the time of operation, and postoperatively. There is one patient here, patient 11, that showed a remarkable improvement. This is called the functional rating scale, which is the subject of intense scrutiny by us and by other investigators to try to figure out why that patient got better. And we're looking into that right now. The forced vital capacity, which is a measure of breathing, is another measure that we can see. And you can see again that there were no major changes or major declines in the slope following surgery. And this is called handheld dynamometry, which is a quantitative measure of strength. And again, in these patients, we didn't see any major changes following surgery. And here's the rest of the handheld dynamometry. We have done autopsies now in all 6 patients who have died, and I can tell you that we have found at least the DNA for these cells in all 6 patients. For the first 5 patients who were male, and the cells that were injected came originally from a male, it was very difficult to identify these human cells in humans. I showed you earlier that it's very easy to identify human cells in a pig because we have specific markers for human cells that the pig cells would not have on them. But when you're putting human cells into a human, that doesn't work anymore, and we have to look for very specific markers. And the ones we're using right now are looking for male markers. These are from the Y chromosome that can be found in a female patient, and we only have 1 female patient. And those cells were found there. The other cells were found based on DNA technology, looking for the donor cells as compared to the recipient DNA. And you can see here, this is one of the spinal cords from the woman patient who died with ALS, and this is the region that we were trying to inject in the circle. And what you can see here, maybe not too well, is that there is an area of clearing here with cells here that don't specifically look like they belong in a normal spinal cord. And here they are right here. These look like the cells, at least, that are injected in. And we can look at these cells. This is what the cells look like before they're injected. And these are very, very similar looking cells that are kind of nested into the spinal cord in this one patient at least. And using those markers for the Y chromosome, we found that these cells did contain a Y chromosome, suggesting that they do not come from the recipient, who is a female. This is the data from the DNA markers, and all of the numbers in black suggest that we have found a percentage of cells that come from the donor that are specifically different from the recipient. And you can see here in all of the spinal cords tested here, at least 5 out of the 6 so far. We now have the 6th one as well. You can see that we found cells, or at least we found DNA from the donor population in all of these, suggesting that even out to 30 months, in patient number 1 who died 30 months after surgery, we were able to find the DNA from the donor cells, suggesting that these cells have survived and probably integrated themselves into the donor nervous system. So the summary and the conclusions. We have completed 18 surgical transplantations in 15 patients. These were 12 lumbar or the low back and 6 cervical, and 3 patients got both. The procedure was well tolerated and safe. We found no indication that the procedure accelerated the course of disease, which was very good news. And we also found that the donor DNA was found in all the autopsy cases, suggesting that these cells don't just get eaten up and thrown away. These cells actually stay in the nervous system, and hopefully they'll do what we expect them to do, which is to nurse the motor neurons that are there and are sick, possibly even back to health. So what do we need to do here? The next phase is to increase the number of injections into the cervical spinal cord and increasing doses, so we think we can create a dose that might in fact be therapeutic for these patients. We have now have our objectives for moving forward. We have shown that surgical transplantation is safe, at least relative to the severity of the disease. And again, this is all risk-benefit. When you're dealing with a disease as horrible as ALS, you're willing to take, and patients are willing to take relatively high risks in order to try to do something about this disease. Again, we need to test whether the disease can slow or halt the disease progression. We've calculated what we consider a target dose, which will require more injections with higher numbers of cells. And again, we'd like to do this with as few patients as possible at the beginning, because we want to try to see if we can make a difference with a dose that we think might be therapeutic, so we can expose as few patients as possible to doses that might be sub-therapeutic. Our current obstacles, we do have funding from the NIH to move forward with the phase 2 trial. When I first gave this talk, we were awaiting FDA approval for our protocol, but since that time, the FDA has approved our protocol, and we are now going through the final stages of preparation in order to recruit our first patients, which we're very happy about, working very closely with the FDA to get this thing moving. We are concerned that the money to conduct the trial will run out if the trial moves too slowly, so we want to move a little bit more quickly. Because a major risk is that we actually won't complete this trial. And if we don't complete the trial, we won't be able to determine if the stem cell injections are therapeutic. And so patients will continue to look usually offshore for stem cell treatments in an unregulated environment. Again, going back to my first point, we don't know if this works or not, and that's why it's called a clinical trial. So we need to move forward as aggressively as is safe, but also with the idea that we're going to end up at the end of the day with an answer about whether this is, number one, safe, and number two, whether it's therapeutic.